All right, welcome back. So what we're going to look at now is the second half or the second part of what we just finished talking about in the first part. So the previous video, if you haven't watched that, make sure you watch that first. That is, we were looking at meiosis 1. Okay, meiosis 1 being the first set of divisions going from a diploid cell to two haploid daughter cells. Each of those daughter cells, again, they're haploids, meaning they have only half the number of chromosomes necessary for a full diploid cell, so meaning they only have 23. And each of those are containing, those two cells are containing replicated chromosomes, so meaning they are a single chromosome for chromosome 1, 2, and so on, but it has two chromatids. Okay, so recall our discussion of the difference between a chromatid and a chromosome. All right. So what we're doing now is we're taking those two cells that are haploid, that have their duplicated chromosomes with the two chromatids, and we're going to further separate those and pull those chromatids apart, and then those chromatids will now become chromosomes again. Right. So this is termed meiosis 2. So this process, meiosis 2, starts in a similar way to meiosis 1 in the sense that we start with prophase, right? So we break down the nuclear envelope, so that membrane surrounding the genetic material. However, in this case, there is no crossing over. So remember, a characteristic of prophase 1, you had that swapping or that exchange of genetic material, right, between the chromosome 1 from the father and chromosome 1 from the mother. There's a little bit of exchange of genetic material. That doesn't happen in prophase 2. Okay, that's already happened in prophase 1. So if you look here in prometaphase 2, you've already got some amount of exchange of genetic material between the two homologous chromosomes that we had in prophase 1, right? So notice here, this is homologous chromosomes. These down here are not homologous chromosomes. Okay, so in prophase 2, no homologous chromosomes, no crossing over, right? So you've got the prophase 1. You go through prophase 2. You go through metaphase 2, so those replicated chromosomes with the sister chromatids are going to align along the middle of the cell, so along that metaphase plate again. The microtubules, those spindle fibers, are going to connect to the kinetochore, just like you had in metaphase 1. And then from the centromere, those microtubules are going to pull that duplicated chromosome. They're going to pull it apart. Okay, so what it's pulling apart are the two chromatids that make up the single chromosome. All right, so you've got one arm, one chromatid here. You've got another chromatid here. You've got the fibers attached, and then they're going to be pulled apart at that kinetochore, right, or at that centromere. So now we've got one chromatid that's going one way, one chromatid that's going the other way, and then when you pull those chromatids apart, you change the name, and now you're a chromosome again. Right, so notice here, anaphase 2, sister chromatids pulled apart, and now you have single-stranded chromosomes separated to opposite poles of the cell. So you go from being a single, large, duplicated chromosome with two chromatids, you pull the chromatids apart, and now you go back to being called chromosomes. Right, so a little bit on, again, the terminology of the terms we're using. And then, like with meiosis 1, we have telophase and cytokinesis. So again, the two chromosomes are now ending up at their respective poles of the cell. It is dividing, right? So that nuclear membrane is reforming. The cell is ultimately pinching off as that cleavage furrow forms. Well, I'll show you the picture of a cleavage furrow in the next picture on the next slide. But essentially, you are separating those two cells, right? And again, these are haploid cells, just like what we finished with in meiosis 1. But the difference here is that the cells produced at the end of meiosis 2 have only a single strand for a chromosome, so a single-stranded chromosome, whereas the cells that were produced at the end of meiosis 1 they were a duplicated chromosome. So it was one chromosome with two chromatids, right? So slightly different in terms of their specific contents, but they're still haploid, right? They're still haploid. So chromosomes 1 all the way through 23, again, even in meiosis 2, they are aligned along that metaphase plate independently of each other, so meaning There is random alignment of the paternal and maternal chromosomes in terms of which side of the cell they face, what pole they face, and so they get pulled in random directions. The X and the Y will move into one, and so if it's a sperm cell with an X that fertilizes the oocyte with an X, then that becomes a genetic female. If it's a sperm cell with the Y that fertilizes the oocyte with an X, that becomes a genetic male. Okay, so again, haploid, 4 genetically different haploid daughter cells is what we produce, right? So this is kind of a summary slide of all of these things happening here in meiosis 2, right? So again, notice what's happening. We are taking our duplicated chromosomes here, right? So notice replicated, duplicated chromosomes with 2 chromatids, right? They're aligning here along that metaphase plate. The spindles are attaching, those microtubules are attaching to the kinetochore. This from the centrosome, the centromere, they're going to pull those fibers, the spindle fibers, back, separate the 2 sister chromatids, and now you call these sister chromatids, now they change back to chromosomes, right? So you call them a chromosome again. So you can look here in the description of this anaphase 2, sister chromatids of each replicated chromosome are pulled apart. Sister chromatids, now called single chromosomes, migrate to opposite pole. Okay, so again, you go from being duplicated, replicated chromosome with 2 chromatids, pull the chromatids apart, and now you call it chromosome. So notice here in the telophase and cytokinesis slide, we're calling these single chromosomes, right? So again, this is what is sometimes confusing for students, is the fact that this cell here, with this one large replicated chromosome 1, replicated chromosome 2, 3, so on, with the 2 chromatids, this is haploid. And this cell here, with a single-stranded chromosome, this is also haploid. Okay, so this isn't diploid. This is haploid. They're both haploid, right? So remember, in meiosis 2, what you start with, and again, you're starting with what you finished with in meiosis 1. You're starting with a haploid cell. Okay, you're just starting with 2 chromatids in the replicated chromosome, and then you're ending up with single chromosomes, right? So a single-stranded chromosome, and again, 23 of those, right? So 1 through 23. The 23rd will be an X in all of them, or I guess technically just the one that's produced in oogenesis, and then in spermatogenesis for males, it will have either an X or a Y. And again, depending on which one of those sperm cells with an X or a Y fertilizes the oocyte with an X, you'll end up with genetic male or genetic female. All right. So this is where we're going to stop for meiosis 2. Please reach out if you have any questions, and I will be happy to answer.